Welcome to Clinical Histology. I'm Dr. Xie from SUMC. In this session, we will focus on the smallest vessels in the cardiovascular system, the capillaries. We are going to talk about the general structures of capillaries, their classification, how they form the capillary bed, and their roles in the microcirculation. First, let's go back to the 17th century. In 1628, William Harvey, the famous English physician, published his greatest discovery, that a fixed quantity of blood traveled in a circle course, formed by the arteries and veins throughout the body. Harvey speculated that a connection existed between the arteries and the veins, but he could not demonstrate this link, possibly because the microscope was not available to him at that time. In 1661, using the newly discovered microscope, Marcello Malpighi, the eminent Italian physician and biologist, described the missing links between arteries and veins, namely the capillaries. Thus, Malpighi completed the chain of Harvey's circle. Now, about 400 years after Harvey and Malpighi's discoveries, we know that the cardiovascular system consists of the heart, arteries, veins, and of course, the capillaries. As shown in this figure, arteries always transport blood away from the heart. They diverge and narrow into arterioles, which will eventually branch into capillaries to facilitate the exchange of nutrients and wastes. The capillaries then join and widen into venules, which will further converge into veins to return blood to the heart. Please note that capillaries are the only part of the circulatory system that permits exchange of materials between blood and the surrounding tissues. Other vessels in the circulatory system are too thick to allow effective diffusion. Next, we are going to see the features of capillary walls, which confer the ability of exchange on these tiny vessels. The diagram on our left represents a segment of capillary. As the smallest blood vessels, capillaries are formed by a single layer of endothelial cells. These cells are joined together by tight junctions and roll up into a tube. The average diameter of capillaries ranges from 4 to 10 micrometers, which permits the passage of blood cells only one at a time. The long axis of each endothelial cell is parallel to the direction of blood flow. The external surface of the endothelium is surrounded by a basal lamina produced by the endothelial cells. Unlike other kinds of blood vessels, capillaries have no other subendothelial layer or tunicus. However, capillaries are usually associated with perivascular cells called pericytes. The pericytes share the basal lamina with the endothelial cells. The well-developed cytoskeletal networks allow the pericytes to contract and regulate blood flow through the capillaries. Furthermore, as a kind of mesenchymal cells, pericytes may undergo proliferation and differentiation, which are important for tissue repair and regeneration. On our right, we have a H&E stand slice of capillaries. Capillary can be recognized by its flattened nucleus adjacent to a small lumen, which is true for both longitudinal and transverse section. Usually, we can find highly eosinophilic red blood cells in the lumen. Please pause the video and try to find out two capillaries in this figure. Can you identify one in longitudinal section and the other in transverse section? Depending on the continuity of capillary endothelial cells and their basal lamina. Capillaries are grouped into three types: continuous capillaries, fenestrated capillaries, and the sinusoid. Continuous capillaries are the most common type, which are found in the connective tissue, muscles, lung, and the central nervous system. Continuous capillaries are lined with uninterrupted endothelial cells. Tight junctions seal the boundary between the endothelial cells to form the intercellular clefts. Furthermore, a continuous basal lamina surrounds the endothelia. Small molecules such as water and ions can simply diffuse through the intercellular clefts or across the plasma membrane. Large molecules are transported by numerous pinocytotic vesicles. That's why many vesicles can be seen in the endothelial cytoplasm. The section on your right shows two continuous capillaries running parallelly to the cardiac muscle fibers, indicated by yellow arrows. Please note the capillary wall and the single row of red blood cells in the capillary. Fenestrated capillaries are very similar to that of continuous capillaries, except that they have windows of fenestry in their endothelial cells. The fenestry are covered by ultra-thin diaphragms of proteoglycans. An exception is the glomerulus of the kidney, which has fenestrated capillaries with no diaphragms. However, the glomerulus has slit pores with a function analogous to the diaphragm of the fenestrated capillaries. Although a fenestrated capillary has windows in its endothelium, its basal lamina is continuous and covers the windows. Fenestrated capillaries are found in organs with rapid exchange of substances between tissues and the blood, such as the mucosa of gastrointestinal tract, endocrine glands, and the renal glomeruli. The picture on the right shows a pancreatic islet. Two capillaries in cross-section are indicated by yellow arrow, and one capillary in longitudinal section is indicated by green arrow. Please note that it is difficult to distinguish fenestrated capillaries from continuous capillaries under light microscope with H&E staining. Sinusoids or discontinuous capillaries are a special type of capillary, which have wide openings in their endothelial cells. Unlike the fenestrated capillaries, sinusoids have no diaphragms covering their large fenestrations, and their basal lamina is also discontinuous. The lumen of sinusoids are more irregular in shape and much wider in diameter. These peculiar structures slow down blood flow and allow red and white blood cells and various serine proteins to easily pass across the capillary wall. Sinusoids are mainly found in the liver, bone marrow, spleen, and certain endocrine glands. This H&E stained slice shows sinusoids in human bone marrow. Please pay attention to the wide and irregular shaped capillaries indicated by yellow arrows. Next, we are going to discuss the capillary bed and microcirculation. As we see in this diagram, an arteria in reddish orange is on the left, and a venule in blue is on the right. The arteria and the venule are joined together by two kinds of connections. In the upper part, we have a capillary bed, and in the lower part, we can see an arterial-venous anastomosis, AVA. First of all, let's take a look at the capillary bed. Capillaries always perform their functions in the capillary bed. As we see, there is a central channel in the capillary bed. connecting the arteria directly to the venule. The proximal region of the central channel is called the meta-arteria, whereas the distal part is called the thoracic channel. Capillary branch from the meta-arteria and converge into the thoracic channels. The meta-arteria supply blood for the capillary bed, while the thoracic channel drain the capillary bed and empty the blood into the venules. The precapillary region, including the meta-arteria, is encircled by smooth muscles, which act as precapillary sphincters. No sphincters are found in the postcapillary region. The precapillary sphincters contract and relax cyclically with 5 to 10 cycles per minute, causing blood to pass through capillaries in a pulsatile manner. When precapillary sphincters are closed, blood flows directly from the meta-arteria and thoracic channel into postcapillary venules. Now let's move to the lower part of this diagram, which illustrates the AVA. The arteria may simply connect with the venule to form the AVA. When AVAs are open, a large amount of blood bypasses the capillary bed and flows through the AVA into the venous system. When AVAs are closed, the blood will choose to go through the capillary bed and finally into the venous system. Capillary beds and AVAs are an important part of microcirculation, which involves blood vessels with diameters less than 100 micrometers and are responsible for the orchestration between circulating blood and the surrounding tissues. The disorders of capillaries, together with the abnormalities of other small vessels, are called microangiopathies. As mentioned above, capillaries deliver nutrients and oxygen to tissue and carry away tissue metabolic products. The more active the tissue is, the more abundant the capillaries. Thus, any disorders of the capillaries may lead to severe malfunction of a specific tissue. One common cause of microangiopathy is diabetes mellitus. Because of high blood glucose level or hyperglycemia, the basal laminae of capillaries in diabetic patients will grow abnormally thicker and weaker, which will compromise their normal functions. Gradually, due to the abnormalities of capillaries, microcirculation will be disrupted and the tissues will be damaged, especially in organs or tissues such as the kidney, retina, and the skin. On your left is an electron micrograph of glomerulus from a healthy kidney, while on the right is a glomerulus from a diabetic patient. Red lines in both figures span the basal lamina. It is easy to appreciate that the glomerular basal lamina of diabetic patient is much thicker. The thickened basal lamina becomes more permeable and less efficient in filtration, which will lead to a steady decline in kidney function. In this session, we have discussed the structures and functions of capillaries. Among all the blood vessels in human body, capillaries have the smallest diameter and the thinnest wall. The wall of capillaries is composed of a single layer of endothelium, which is encased by basal lamina. Pericytes are frequently found to surround the capillary wall and share the basal lamina with endothelium. Pericytes play critical roles in the development and physiology of capillaries. We also classified the capillaries into 3 groups: continuous, fenestrated, and sinusoidal. They differ in their structures, functions, and locations. Capillaries always perform their functions in capillary bed. The capillary bed, together with other small blood vessels, constitute the microcirculation in organs and tissues. Blood flows through microcirculation to distribute nutrients and oxygen to the tissues and take away the metabolic products from the tissues with extremely high efficiency. That's all for today. Thank you for joining us. See you next time.